Hello, my name is Britta Peel, and in this lecture I will be discussing the role of the pancreas and liver as accessory organs in digestion. They are classified as accessory organs, meaning that they lie outside the digestive tract, but they contribute to the overall process of digestion, as we will see throughout this lecture. So up first, I will be discussing the role of the liver. The liver is the largest organ in the human body, weighing approximately 3.3 pounds in an adult. It lies on the right side of our body, just under the diaphragm. So here you can see the lungs, and highlighted in red here is the liver. And it's known as the biological filter of the body, so it filters all of our blood, and we'll discuss that a little further. It also has a crucial role in fat digestion by producing bile salts, and we'll discuss that as well. And quickly, I just wanted to note that it produces the hormone hepcidin, which is secreted, and it leads to a decrease in iron absorption. So when we have high iron content in our blood, the liver will secrete this hormone and subsequently lead to a decrease in absorption. Okay, so the hepatic portal system is the part of the liver that I referred to as the biological filter. So hepatocytes, those are liver cells, contain a variety of enzymes, and such as cytochrome P450 isozymes, and they metabolize drugs and xenobiotics and clear them from the bloodstream before they reach systemic circulation. So as we can see here, the large and small intestine is covered by a variety of capillaries, so as nutrients are absorbed into the bloodstream following digestion, they first pass through the hepatic system directly to the liver, where they are filtered, and then back into the hepatic vein, which truncates into the inferior vena cava and goes back to the heart. So it allows for filtration before we have blood going to the systemic circulation. And this is a really important part of the liver's role, and it really allows for us to clear toxic substances that we might ingest. It's a part of our primary defense. Okay, so next we're going to discuss the liver's role in fat digestion. So hepatocytes, that's again liver cells, secrete bile, which is a non-enzymatic solution, which is comprised of three main components. The first being bile salts, which are detergents that emulsify fat droplets to make them more soluble and increase the surface area of fat, allowing for digestion in the small intestine. Also in bile is bile pigments, which are composed of waste products of hemoglobin degradation that occurs in the liver. So it's a way that we also excrete waste products of our metabolism. And our third component is cholesterol, which is again is excreted in our feces and other drugs and xenobiotics. So they're being cleared from the blood in the liver and secreted in the bile. And they will not be reabsorbed, so they'll go out our digestive system through expulsion. So the bile is produced in hepatocytes, and it travels via hepatic ducts to the gallbladder, which is seen right here, for storage. And the gallbladder will contract following the ingestion of a meal containing fat, which allows for bile to be released into the duodenum, which is the first part of the small intestine here, through the common bile duct. So it'll be excreted from the gallbladder into the duodenum, where it is able to emulsify those fats. So bile salts are amphatic, meaning that they have a hydrophobic and hydrophilic component to them. The hydrophobic is attracted to the fats because those are hydrophobic substances, meaning they're nonpolar. And it's also hydrophilic, which is attracted to water. So it's allowing for fat to be separated. And then colipase, which is a enzyme that is released from the pancreas, which we'll talk about in an upcoming slide, actually displaces some of the bile salts, allowing for lipase access so that the fats can be digested. So it further increases the surface area of those fats, allowing them to be digested by enzymes in the small intestine. But without bile, we couldn't digest our fats because they would remain in big groups and wouldn't be able to be separated into droplets that is seen thanks to bile. Okay, so next we're going to discuss the role of the pancreas. So the pancreas is found deep to the stomach in our abdomen. So here it's highlighted in pink, and then above this is the gallbladder that's highlighted. And then the common bile duct that connects the two, and then it's connected here to the small intestine, the duodenum. So the pancreas has a endocrine function, meaning that it releases hormones into the blood, and that is from clusters of isolate cells. Beta cells produce insulin, which is released in response to high blood glucose levels, signaling to our cells in our periphery to uptake glucose. And it also, isolate cells also are alpha cells, which produce glucagon, which is a signal that the glucose is gone. So it signals to our cells to metabolize glycogen stores as their primary source of ATP. So the pancreas also has an exocrine function, which is more in tune with the digestive portion of it. So we'll discuss that in the upcoming slides. And that function is from Acini lobules in the pancreas, and they produce digestive enzymes and sodium bicarbonate, which both play a major role in digestion. Okay, so on this diagram, you can nicely see the isolate of Langerhans cells. So those are the alpha and beta cells of the pancreas that produce insulin and glucagon, respectively. And here is the Acini lobules. You can see, and they are the exocrine portion of the pancreas. So exocrine meaning that they produce substances that will be secreted via a duct to the target tissue. So that's here. And this duct is controlled by the sphincter of Oddi, which controls the release into the duodenum. So the acinar cells produce a variety of enzymes that are secreted as zymogens, meaning that once they are secreted into the duodenum, they must be activated. Their release is stimulated by psilocystokinin, which is produced in the stomach in response to digestion occurring there. So this will then produce it will cause the acinar cells to produce these zymogens and secrete them into the duodenum. So the first one secreted is trypsinogen, which is then activated by the brush border of the small intestine to its active form trypsin, which acts in the digestion of protein, and it also acts to activate the other zymogens secreted by the pancreas. So chymotrypsin in its active form, chymotrypsinogen, and then its active form chymotrypsin, which also contributes to the digestion of protein, and then procarboxypeptidase, its active form carboxypeptidase, which digests protein again, procolipase, which then is activated to colipase and is active in the role of lipid digestion, which is what we discussed earlier, assisting bile to increase the surface area of those fat droplets. And then prophospholipase, which is activated to phospholipase and also aids in the digestion of phospholipids in the small intestine. So all those enzymes are produced by Acini cells in the pancreas and secreted into the small intestine, where they contribute to digestion. So lastly, we will discuss the production of sodium bicarbonate by duct cells of the pancreas. So sodium bicarbonate is secreted by duct cells into the duodenum, stimulated by the secretion of secretin. So S cells of the duodenum produce a enzyme or a hormone known as secretin, um, due to the presence of acidic chyme that is entering the small intestine, which then causes duct cells to release sodium bicarbonate. So the secretion of sodium bicarbonate is a passive process that occurs via electrochemical gradients and osmotic gradients. So this is a complicated diagram, and I'm going to attempt to explain it. So the sodium potassium chlorine cotransporter, seen here, transports chlorine, potassium, and sodium into the duct cell, which then allows for a chlorine gradient. So chlorine is then transported out of the cell with the cystic fibrosis transporter, out of the cell into the lumen, to the apical side of the cell, the lumen of the pancreas. And with that transport, also sodium bicarbonate is transported out. Duct cells have a high concentration of an enzyme known as carbonic anhydrase, which is what produces bicarbonate in the cell. And it's also important to note that the sodium potassium pump is working on the basolateral side of the cell to create these gradients that allow for this transport. And interestingly enough, the hydrogen that is produced by carbonic anhydrase is leaving the cell on the basolateral side in an exchange for sodium that helps to neutralize the basic characteristics of the blood as a result of parietal cells' production of hydrochloric acid in the stomach. So that's a really interesting part of this pathway. So this hydrogen is actually neutralizing the blood that is traveling to the pancreas from the stomach, and then it's going to go to the liver. So yeah, CFTR, this transporter up here, is really important, and people that suffer from cystic fibrosis have a defective transporter here, and therefore there is no transport of chlorine, and then there is no transport of bicarbonate. So they have a thick mucus that accumulates in their pancreatic ducts and clogs them, which is unfortunate, and they receive treatment to try to neutralize that. But once bicarbonate is in the lumen of the pancreas, it is then secreted into the duodenum, where it neutralizes the acidic chyme in an acid-base reaction that is detailed here on the slide. So that concludes my lecture. Thank you for listening to me. I apologize for my ums and okays throughout. This was an interesting assignment and one that I haven't done before, but it was fun and helped me learn a lot. So thank you.